It's really my pleasure to be here today to tell you about some of the work we do at ELSI. I'm going to cover the topic of how we go from the origin of life to something a little more complicated that we think could have been the ancestor of all the living things we see on the planet today. I am a chemist, so I tend to think of this in chemical terms as a chemical problem, so bear that in mind. So the central question we're trying to understand, we heard a lot of talks this morning about how planets form and how the atmosphere and oceans originated, but how we went from a primitive Earth that had no life on it, it was just rock and water and gas, to the kind of planet we live on today, right? Which is just covered in living things, of incredibly complex ecosystems, all kinds of amazing organisms that are doing interesting things to stay alive and make copies of themselves. In the oceans, on dry land, tropical jungles, and even if we get down to the microscopic level, there's an incredible diversity and richness of life at the microscopic scale, right? It's just very complex. So the question is, how did we get to that from this lifeless state? And it's really quite a vexing problem. So Charles Darwin, when he was formulating his theory of evolution by natural selection, came up with the idea that organisms change through time, and an ancestor organism can give rise to, say, two different types of organism, right? But if you reverse that and go backwards in time, the implication is that all these organisms at the tip that are alive today have a common ancestor, right? And in fact, they should have a single origin if they're all related, right? And he wrote that in the first edition of his book, very eloquently. There is grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one. Right? And then once you have that first thing, it is free to become many new things, right? But this question of how you make that first thing is very complicated, right? We have a general scheme for how we think this process occurs. And what we think happens is we have some sort of chemistry that takes place before you have living things. This is chemistry that the environment does. I'll show you some examples of that. There's some process here that we don't understand very well at all that gives rise to a first living thing. That first living thing may be very different from living things as we recognize them today. But it goes through some period of evolution until it gave rise to what we call MUCA, or the last universal common ancestor. And that putative organism is the thing that gave rise to all of the species we see today, okay? And, which is contemporary biochemistry. So things we don't know, what kinds of chemicals are necessary for this process, and how those chemicals come together to make living systems without the assistance of other living systems. There's two ways you can think about this. One is a sort of bottom-up approach. So if you're a chemist, you say, okay, what chemicals do I have? How can I put them together to do interesting reactions? If you're a biologist, you can do what's called a top-down approach. You can say, what sorts of things do all modern organisms have in common? And if you say, okay, only the ones that they have in common are the crucial ones, which ones of those can we cut away? Can we design a simpler first organism? Right. So you're comparing modern biology. Okay, so we know a little bit about when we think this happened. We can date the Earth pretty well at this point. We know the Earth is about 4.5 billion years old. So we're going back in time along this axis. We are here, right? This is the present day, 0 years in the past. If we look at the geological record, we have evidence for biology going back about 3.5 billion years ago. It's quite remarkable, but we do see things that we can agree are evidence of living organisms. We also have some indication of when there was liquid surface water on the Earth. So we think all living things need water. So the planet had to cool enough for that to exist. And what that leaves us with is about a billion-year window for this process of life to originate, right? And it doesn't mean life didn't start earlier, we just don't have evidence for it, okay? And really the big question is, we don't know if this process takes a week or a billion years, right? It could be that it's just something that takes an incredibly long amount of time. It will be very difficult to observe in a laboratory. So this is a fundamental unknown. Okay, so just to give you some context for why it is so difficult for us to know when life started, because the Earth is such a dynamic planet, we heard today about the process of tectonics, that rocks are constantly being made in these, they're called Midocean Ridges in the middle of the ocean. We have new rock coming up from the interior of the Earth, cycling out like a conveyor belt, and eventually being pushed back down into the Earth. In fact, all of this rock is fairly young. Maybe the oldest oceanic crust is 400 million years old, right? So it's not very old at all. The rocks that make up the continents can be quite a bit older, but these little regions in purple here are the oldest, large domains of rocks we know of. They're only about 3 to 3.5 billion years old, okay? So the point is, because the Earth is constantly doing this recycling, the oldest rocks really aren't available to us, which are the rocks we would be the most interested in. There are a couple of regions where we have rocks that are almost 4 billion years old, but they are heavily, they've been heated, they've been put under pressure, and we don't think that we're going to be able to find evidence of life very easily in these kind of rocks. Okay, so again, switching to the sort of top-down approach, we can look at what we think is the simplest form of life that we see today, and something like a bacterial cell. Actually, it's a pretty sophisticated little machine that's function is making more bacteria, right? And if you're not familiar with the structure of a cell has a cell membrane. It is a wall that encloses the contents of the organism. It has a genetic molecule called DNA, which stores the information for making copies of the cell. And it has various small molecules that help integrate the functions of these different components. All right, and here's an electron micrograph of a bacterial cell, just so you get a sense. Unlike a machine that you're more familiar with, all of the parts are mixed together, right? They work even though they're always diffusing around and moving in space and time. Okay, so just to give you some appreciation, if you can play this video, this is approximately real-time animation of the process by which DNA in our bodies, in bacterial bodies and all living things' bodies, gets converted to another molecule called RNA. So it's this copying of information by a series of protein enzymes, right? So you're gonna see this protein enzyme coming here, it's moving along the DNA, which is the red strand, and it is copying an RNA molecule, which is a yellow strand. This is approximately how fast this occurs in real time in your cells. So this is all going on right now inside of us, very quickly. That RNA strand then gets picked up by a rather complicated other machine within the cell called the ribosome, which takes that information using adapter molecules and turns it into a third kind of polymer called a protein, all right? So you'll see in a second here, there's the protein starting to emerge from this large machine. So this is a very complicated process, right? There's a way of translating from one language to another language to a third language very precisely and very quickly, all right? It's very difficult to understand how a process that complex could have arisen by itself. So we think we're looking for simpler types of chemistry that eventually became more complex over time. So there was a Hungarian theoretician named Tibor Ganti who came up with this notion of, you can think of a cell in a very idealized way as just being 3 systems, okay? One of those systems is a container that keeps everything together, right? One of those systems is, again, some sort of metabolism or machinery for making the parts. And the third system is something like a blueprint. It contains the information for making the other parts, right? And those things all have to be connected together in order for the whole system to propagate itself together. But the important message here is maybe you could make these kind of machines, these kinds of systems, out of completely different types of chemicals than modern biochemistry is made out of. Right? And indeed, we might expect if we went to look for life on other planets, it might have evolved into a pretty different configuration than the way our life is. So let's think a little bit about where we get the chemicals from to start this process. So space is one source. We have things like comets and meteorites that are always coming in. So it's actually an image captured from a dashboard camera on a car of a meteorite that exploded over Russia a few years ago. These kind of processes are happening today. We know something about the contents of these materials in terms of chemicals. We believe these things were happening more frequently on the early Earth. Another possibility is that storms and sorts of processing of the Earth's atmosphere generated different kinds of chemicals. It's a view of lightning storms from space, these little purple flashes. Again, there's lightning on Earth all the time. We believe that's been happening for billions of years. And you can get some very interesting chemistry taking place in the region right next to a lightning bolt. It's very hot. It's able to do interesting chemistry. And there's a third possibility. And these are not mutually exclusive. In fact, because the Earth is releasing heat from its interior, that heat comes into contact with ocean water and rock deep in the Earth's surface. So it's sucking ocean water down here, comes into contact with hot rock, and it gets ejected as extremely hot water that is also able to do very interesting chemistry. Right? So there's a lot of ways we think we can make chemicals on a primitive, lifeless planet. Just to give you an example, I actually do work on meteorites as part of my job. This is not my personal work, but we do this kind of thing. You can take meteorites and you can extract the organic chemicals from them and analyze them. And there's a device called a mass spectrometer, which essentially allows you to weigh the types of chemicals that are in it. And what you end up with, this is an example of a meteorite that fell in Australia. There are thousands and thousands and thousands of compounds with distinct weights. Okay, so they're all different types of organic compounds. And just as there might be more than one person in this room who weighs, say, 75 kilograms, there may be multiple chemicals that have the same weight. So in fact, this is probably tens of millions of distinct types of organic compounds. Right? Very, very complex chemistry going on in our solar system. Another example, this is something you can do in the lab. If you put some simple gases like methane, ammonia, and water in a flask, and we pass a little electric discharge here, so there's an electric discharge generator, it's a spark simulating lightning. After a couple of days, that gas gets turned in, again, to a very, very, very complex mixture of organic compounds, right? This is something you can witness over days in the laboratory. And it's simply because in that electric discharge, these molecules get ripped apart and recombined and then undergo more complex chemistry that eventually gives rise to, again, millions of different types of organic compounds. So how do we put those things together to actually make a living thing? That is a much more complex question. Yeah? One of the things we think might be really important in this process, in a very general sense, is something in chemistry that we call autocatalysis. All right? And autocatalysis is simply this. When a chemical reaction, you can think you have things that you mix together and you get a product, right? In an autocatalytic process, the product of the reaction actually goes back and speeds up the reaction, right? So it's a catalyst for its own production. And if you look at the amount of product over time, it actually increases and increases over time, right? The more C you have, the more of it you can make, which gives you more C, which means you can make more C, and so on and so forth. There are different ways you can think about doing this, but that basic process of amplification through autocatalysis may be a very important thing in biology. And if you want to think about that in biological terms, you take carrots and you give it to rabbits, you give them to rabbits and you get more rabbits, which can eat more carrots to make more rabbits, to eat more carrots to make more rabbits, and so on and so on. So that is it feels like a very biological process to us, yeah. We have different ways we can simulate these sort of processes in computers, so sort of reaction diffusion processes, where you can actually have two of these systems coupled, where the product of one reaction becomes the food for the next. And you can see that you can get very complex sort of lifelike behavior in a computer from these very simple reaction models. Yeah? So things that seem to make copies of themselves by converting food into their own substance. So I'll give you one more kind of real-world example. This is, so we take two simple molecules, HCN, hydrogen cyanide, and formaldehyde. It's HCHO. One has 3 atoms, one has 4 atoms. Put it in water. We're going to look at this reaction under a microscope. All right, this is about 2 millimeters across. We're speeding this up to about 5 hours. The molecules are smaller than you can see with a microscope. But over time, you'll notice, you start seeing some little specks forming here. And it gets faster over time. And it gets faster and faster over time. You start seeing more and more and more and more of these, till it turns into a complete snowstorm of something being formed, small particles. Essentially, it seems to be an autocatalytic process, and it's a very interesting one. I'll show you a little more about it. You start aggregating more. Until at some point you start running out of the starting chemical and it slows down. Until you have these very large and complex sort of structures that form. And you could take those things and look at them under a higher power microscope, and you see, in fact, that what they are is small, kind of cell-sized little capsules of chemicals. All right? They're hollow. You can put stuff inside them. When you put them under the high pressure or the low pressure of the electron microscope, they explode, sort of like the eggs in the Alien or so. You know, they rupture. You can drill holes in them. We know they're hollow. So there are these very interesting chemical phenomena that we don't understand very well that might be relevant to this sort of process. They're autocatalytic. They use very simple chemicals to become more complex things, so on. We're trying to understand the chemistry of these kind of systems, which can be quite complex. In this case, this does seem to be an autocatalytic process. It goes around, it's cyclic, it puts out a large molecule as a product. So you can think of the HCN as being the food in this case. It feeds in at 4 places in the cycle. The aldehyde is a catalyst. It gets regenerated in every turn of the cycle. That thing outputs these little ball-shaped things. And the ball-shaped things themselves seem to be a catalyst for the overall process. So this is a form of two types of nested autocatalysis. Yeah. So where does that leave us? We really don't know how life started or evolved to give the progenote. We have a lot of models. We can test things in the laboratory, see which ones are better and which ones are worse. We know something about the timing, plus or minus a billion years. Yeah? We know something about the types of chemicals are involved. Not everything, not as much as we need to, certainly. We know something about self-assembly chemistry. Not everything we need to know, but something. There are many, many, many very mysterious problems left to be solved in this field. So hopefully we'll make some progress here. And I lastly just want to thank you all for coming.